Dear friends, I will discuss some of the questions relating marked and release and some regarding Agrobacterium tumefaciens. This is the formula. R upon T is equal to M upon N, where R is the marked and recaptured and T is the total in second sample equal to marked in initially and N is the total population size. Suppose some research scholar went to the field and that scholar captured 100 fishes from the pond. So, and he marked 100 fishes, then 100 upon N is the population size. And again, he visited the area and again he recaptured 150, and out of which he had only 70 marked. So 70 is equal to R and T is equal to 150. So 70 upon 50 is equal to 100 upon total population size, just like this one. So this is the question. In order to estimate population size of a fish species in lake, a researcher captures 100 fish from the lake and marks them with the color tags a week later, means 100 upon total population size. And a week later he catches 150 fish, of which 25 were then tagged once. Okay, so 25 upon 150 is equal to 100 upon total population size. This is the one. So answer should be third, 600. In next question, you will see a grasshopper population is being assessed by capture and mark release recapture method. On the first day, 100 grasshoppers were captured from a given area in 1 hour time, marked and released. On the next day, during recapture, 10 marks and 90 unmarked grasshoppers could be found in the same time period from the same area. What will be the estimated population size in the given area? Again, so it will be 1000, because in 100 it is the first day recapture. 100 upon total is equal to 10 marks, 10 upon 100. In this case, in a census for the lake, 10 individuals were marked and released and sampling were taken. So 10 upon total. After a few days, 15 individuals were caught, of which 5 upon 15. 5 upon 15 is equal to 100 upon total. Answer should be 30. This is the question of December 2019. Very good question. Because mark and recapture is never %. Okay. So you can solve this question after following the formula given on the first slide. Okay. Now these are some material regarding Agrobacterium tumefaciens. Okay, Agrobacterium tumefaciens is the gram-negative bacteria that cause ground gall. The tDNA, transferred DNA, which at least contain two sorts of the genes. One is oncogenic genes, which is tumor causing, that encode protein required the combination of auxin and cytokinins, and the other genes required for the opine. Opine is the non-protein amino acid production. So both genes are present on the T plasmid. Okay, both tumorigenic and opine catabolism gene are found inside the tDNA of the Ti plasmid, though the virulence genes are situated outside the tDNA on Ti plasmid and bacterial chromosome. Okay, these virulence genes are available onto a few operons, and these genes are the wire A, wire B, wire C, wire D, wire E, wire F, and wire G and wire H on Ti plasmid. And the different operons CHVA, CHVB, CHV, these are found on main chromosomes. These are not situated on the Ti plasmid, but these are required for the infection of Ti plasmid. Okay. The transformation procedure begins with the connection of the bacteria with the plant cells and took after by the stimulation of the expression of wire A or wire G. So initiation started with the wire A and wire G. Okay, then wire D2 is attached with the 5' end of the Ti plasmid, and wire E helps in the transfer of this one because it protects the T plasmid complex from degradation of host enzymes, and wire B and D4 types, these form the channel through which the Ti plasmid migrates into the host cell. Okay. Wire E2 protein are the single-stranded DNA restricting agrobacterium proteins which are shipped into the host plant cell where they shield the mature T complex from degradation while traveling to the host nucleus. A DNA-like Arabidopsis protein DLC3 combined with the another protein VIP1 which is required for the coordinated developments in the intracellular transport of agrobacterium into T complex. VIP2 protein interacting proteins has been exhibited to assume a noteworthy part in tDNA integration into the plant genome. Wire F, it is associated with the degradation. It is wire F to target wire E2 and VIP1 for degradation in each cells and thus advancing the destabilization of VIP1 in the host cell. So wire F for proteasome. One question came, this one. Okay. So wire D, wire G, wire D2, wire B1 and wire F. So wire G is for the induction of genes, means 4-1. And wire F for proteasome, that is the third one. And wire B1, the components of the transfer complex. Okay. And this one, the wire D2, nucleate transfer of the DNA, because wire D2 it attached on the 5' site of this one. Okay. So these are the some nitrogen fixation genes. These are the capital NOD genes and small NOD genes. Capital NOD genes means host genes. These are the nodulin genes. Plants genes specific to nodules are called as nodulin genes and expressed as the capital NOD. Small NOD genes, these are found in the bacteria. Rhizobial genes participate in the nodule formation and are called as nodulin genes. And common node genes, these are the node A, node B, node C and node D. Out of which node D is the regulatory, while node A, node B and node C, these are the metabolic type. They produce enzymes. The node D is constitutively expressed and its protein regulates the transcription of other node genes. Okay. The protein of node D attached on the node box. The host specific node genes, node P, node Q, node H and node F, node E and node L. These are the host specific host specific node genes. Node P, these are the just qualitative type. These are not compulsory, but node A, B, C and D, these are the common to all rhizobia. Okay. Isoflavonoids and betanes secreted by the roots, these attractants activate rhizobia node proteins, which then induces transcription of the other node genes. These promoter regions of all node operons except that of the node D contains highly conserved sequence called the node box. Node factors are lipochitin oligosaccharide signals, node molecules, all of which have a chitin B14 linked uh, N-acetyl D-glucosamine backbone varying in length from 3 to 6 sugar units and a fatty acyl chain, the C2 position of the non-reducing sugar. In December 2019, in June 2018, one question came and that changed the alpha 1,4 link for this one enzymes. Okay, so node A enzymes is the N-acetyl transferases that catalyze the addition of a fatty acyl chain. Node B is a chitin oligosaccharide deacetylase that removes the acyl group from the terminal non-reducing sugar, and node C is a chitin oligosaccharide synthase that links the N-acetyl D-glucosamine monomers. Okay. Again, this is very important slide, and this could be the question. And after this slide, I will dispatch you some questions of relating with the nitrogen metabolism. Okay. So I think that will help you in tomorrow exam. Good luck.